Cardiac-derived cells are placed onto cell culture plates for a simulated ischemia reperfusion model. Ischemia is achieved with 150 minutes in a glucose-free medium while the oxygen level is below 0.5%. This ischemic period leads to the shrinking and blebbing of most cells. Then, normal media and oxygen levels are reintroduced to simulate reperfusion. Mesenchymal stem cells are added and cultivated together with the damaged cells. After 24 hours of co-cultivation, cells are stained with calcein and ethidium-homodimer to differentiate between live and dead cells. Confocal microscopy clearly shows the two cell populations and their interactions. Flow cytometry analysis reveals 3 clusters of damaged cells which can be plotted on a graph and analyzed statistically.
Stem cell transplantation protocols are finding their way into clinical practice, getting better results, making the protocols more robust, and finding new sources for implantable cells are the focus of recent research. Investigating the effectiveness of cell therapies is not an easy task, and new tools are needed to investigate mechanisms involved in the treatment process. We designed an experimental protocol in order to allow the observation of cellular connections and even subcellular mechanisms during ischemia reperfusion injury and after stem cell transplantation. Our design criteria were fully in vitro system where only the host and the donor cells are present, so it is isolated from the surrounding tissue. Continuous measurement of cell viability before, during and after the injury for up to 24 hours. And clear identification of both cell populations so the fate of the grafted cells can be traced. In order to reach this goal, we set up a protocol of oxygen glucose deprivation, which is commonly abbreviated as OGD, followed by a reoxygenation period. Our choice of host cells was H9C2 cells, a well-known red cardiomyoblast cell line. After OGD, the damaged cells were supported by transplantation of bone marrow-derived mesenchymal stem cells by adding them to the culture. This setup allowed us to investigate the intercellular connections using confocal fluorescent microscopy and to quantify the survival rate of damaged cells by flow cytometry. Let's get to work and see how we do it.
Remove culture medium from H9C2 cells. Wash cells twice with PBS and digest with 0.05% trypsin EDTA. Place petri dishes for 5 minutes in a 37-degree incubator. Inhibit trypsin with DMEM culture medium. Spin down the cells at 1200 RPM for 8 minutes. After removing supernatant, resuspend cells in 1 mm DMEM culture medium. Count cells with hemocytometer using trypan blue staining. See 30,000 H9C2 cells per well in a 12-well plate. Place the plate into the 37-degree carbon dioxide incubator for 24 hours. Remove medium by aspiration. Wash cells twice with PBS. Add 3 mm glucose-free medium. Place the plate into the cell incubation system. Start nitrogen gas to purge the oxygen out of the incubation system. Wait until the level of oxygen reaches 0.5%, then start the timer. 0.5% oxygen means approximately 3 mm of mercury partial tension. Subject cells to 150 minutes of simulated ischemia. During ischemia, cells are getting detached from the surface and their membrane starts blebbing.
We found that each cell population needs slightly different lengths of time until reaching a 60 to 80% of cell damage. That's why we suggest to begin with a 2-hour incubation, and if it's not enough, increase the time of OGD. In our case, H9C2 cells typically need 150 to 70 minutes of OGD.
At the end of simulated ischemia, remove the glucose-free medium from the cells. Pipette normal culture medium to the wells. Place a 12-well plate for 30 minutes in the 37-degree carbon dioxide incubator. This is the reperfusion period. During simulated ischemia, prepare rescue cells for transplantation. Dilute the fluorescent dye vibrant DID in 1 to 200 ratio in DMEM culture medium. Aspirate medium from the rescue cells and add 300 microliters DMEM from vibrant. Place petri dishes for 30 minutes in the 37-degree carbon dioxide incubator. After 30 minutes at the end of the reperfusion, add 20,000 mesenchymal stem cells per well to the postischemic H9C2 cardiac myoblasts. The initial steps are the same as H9C2 seeding, washing 2 times with PDS, trypsinization, and counting. Place co-culture of cells into the 37-degree carbon dioxide incubator for 24 hours. After 24 hours, harvest the culture media and the adherent cells from wells with trypsinization. It is very important to collect the culture medium also because some dead cells could have detached from the culture dish surface during the 24-hour incubation. Suspend the cells in 500 microliter live dead dye solution, 5 nanomole calcein and 400 nanomole ethidium-homodimer in PBS, 5 times 10 to the power of 4 cell per milliliters. In case of co-cultures, mesenchymal stem cells are distinguished from H9C2 cells by the respective fluorescent cell labeling. This way it is possible to detect cell fusion and cell-to-cell contact sites. The ratio of dead cells can be evaluated in 4 independent fields of view using a 10 times objective for each culture in a blind fashion, which gives a reproducible cell count result. Transfer cells to flow cytometry tubes. Start the CellQuest Pro software. To set up the flow cytometer for measurement, prepare control samples of live and dead cells. Living cells are cultured at the same density in normal conditions and stained with calcein. Dead cells are prepared with 1-hour treatment with 10 micromole hydrogen peroxide, stained with ethidium-homodimer and harvested in the same manner. The instrument settings should be adjusted so the living cells are calcein positive and ethidium-homodimer negative, while the dead cells are calcein negative and ethidium-homodimer positive. The percentage of these populations can be expressed as bars on a graph and statistical analysis can be performed on these values.
We found that the addition of healthy stem cells increased the ratio of surviving cells in the population. Interestingly, we also observed that the cells which were saved after the injury can be gated out from those which were dead, but also from those that remained alive. We believe that this population warrants further investigations.
After watching this video, you should have a good understanding of how to conduct simulated ischemia experiments with in vitro stem cell transplantation in a standardized way. So that's it. Thanks for watching and good luck with your experiments. In case you have any technical questions, do not hesitate to contact us. We will be there to help out.